Now it's time for Marcus from Amniotics. Marcus Larsson, please go ahead and present Amniotics.
Thank you very much. So, good day. My name is Marcus Larsson. I'm the CEO of Amniotics, a stem cell company based in Lund. My background is from the clinical side. I'm a consultant in pediatrics and neonatal intensive care. And I've also a research career in clinical physiology. This was the basis for the discovery of a novel type of stem cell found in amniotic fluid, the fluid protecting the fetus, and this just prior to birth. Amniotics was founded as a university spin-out in 2015, listed on Nasdaq First North Growth in 2021. We are based in Medicon Village, and we are about 8 full-time employees. We develop drug candidates for cell therapeutics, both as a platform and as a lead clinical candidate. We are in clinical phase and have completed a phase 1b study to determine the dose of choice, and are now planning a phase 1 study in our target indication. We also have an emerging platform technology in the IPS field, where our particular interest is to work towards allogeneic NK cells. We have an in-house GMP facility with the capacity to produce cell therapeutics for clinical trials, and this gives us the advantage, as other stem cell companies have seen too, to control the development process, making it GMP compliant, but also being able to optimize cost of goods and being agile. We are also investigating and driving having a contract development and manufacturing offer to clients. So, what are we working with? We are working with a biological material which is normally discarded. So we have developed a technique and a patented medical device, which is class 2, that we produce, that safely for mother and child can extract the amniotic fluid during planned cesarean sections, which is about 20% of deliveries. We do not need to take all of these deliveries. We only need a few. From each collection, we can scale the material we collect to 6,000 treatment doses currently. And we can also use this harvested neonatal quality material for future development of cell therapeutics. So what is interesting here, and this is the core of the concept, is that in utero prior to birth, the child has slightly higher water content than we as adults have. And this makes the child or the fetus to soon be born leaky. So when we first looked at this, we studied the amniotic fluid, but did not think that there would be any living cells there. We thought, okay, there will be dead skin cells shedded off. But to our and also this material is recirculated every 72 hours, so it's a sort of a fresh pool of material. And to our surprise, we found a lot of viable cells in this material, and these cells turned out to be mesenchymal stem cells. And even more interestingly, there were 4 subclasses. So from different anatomical compartments, these cells tend to leak out. So they leak from the skin, they leak from the lungs, the baby urinates out into this system. We have a nephrological type of cell, and also leaking from the central nervous system. And that is the basis for this platform, that we have 4 different cell types. We select these cells and in a GMP fashion. We have done genomic studies to understand where these cells come from and can pinpoint these populations. We scale them up and we package them in cryovials at -150. So our development pipeline so far, we have completed as some other companies also did, explored cell therapeutics during the COVID pandemic. And all of these, when you plan these studies, this was prior to the vaccines. And basically, we all know that the mRNA vaccines in particular has made, and also antivirals, has made COVID a non-interesting target for an expensive cell therapeutic. But we did get valuable safety data and dose data from the study. And it was a first in man. We are the only ones in the world working with full-term amniotic fluid cells. So we have turned our interest to an area which fulfills the important criteria for a cell therapeutic. And what are they? It is that the severity of the disease warrants to give biological material from another person. I mean, this is complicated. It's similar to a transplantation. It is expensive to do these cell therapeutics. We are working to reduce cost of goods, of course, but still, it's costly. And most importantly, you need to have, as a company, extremely solid preclinical data and clear effects to warrant going further in the exploratory approach. And there are also, regarding the source material, you have to have control of it. You have to be able to do it rather cheaply, bring down cost of goods, and it must be ethically sound. And finally, it's preferable to have neonatal quality of the cell material you give. And we also have programs in oncology, a project that has EU support in doing allogeneic NK cells. This is a discovery program aiming to, within 3 years, go into a preclinical stage. So this is a discovery program, and we have other more dormant legacy programs in the other cell types, which we, out of priority reasons, funding reasons, are letting lie for present. So the phase 1b in COVID is completed, and we are turning to lung transplantation. And I want to tell you a bit about this. Why are we going into this field? Well, there is, of all the organ transplants, lung transplantation has the poorest outcome. So after 5 years after receiving a new lung, half of the recipients are dead. This is very, very poor outcome. The sort of an early harbinger of this is a condition called primary graft dysfunction. It comes during the first 72 hours, and it indicates early sickness and death, and later sickness and death. So the goal is to here increase survival, but also to a long-term goal to, via cell therapeutics, alter the ecosystem around transplants, how we maybe pre-treat the transplants before putting it into the recipient. So this is primary graft dysfunction after lung transplantation. Basically, the lungs get more cloudy, get more fluid in them. You notice this with higher need for oxygenation externally and bad blood gases. And it's graded 1 to 3. And the grade 3 condition affects about 30% of those receiving a transplant. So here we come to the sort of preclinical reason why you want to move ahead. So we did a study with a key opinion leader in transplantation, and the best model is to use a large animal model, pigs. So 12 pigs were induced with the primary graft dysfunction, a very complicated model, a 3-day session with full operating team. And in the non-treated group, 5 of the 6 animals in the control group had the severest condition, and 1 had the second most severe. And in the treatment group, there were grade 0, no PGD. And the blood gases, as you see, or the relationship between how much oxygen you give and how much is in the blood was significantly more favorable in the treated group, and also with lymphocyte counts in the lungs. This is the kind of data, like almost binary in style, that we need to want to progress. So this is our target indication. There are about 5,000 transplants in the US and EU. It's a high cost maneuver. It's high risk for the patient. There is room for improvement. And this is a field where we think that it's warranted to do the study. The healthcare system needs it, the patient needs it, and we can sort of get back the costs necessary as a company. We also have a program in CNS called Cognistem program. It's one of our early programs, but I mention it because it's interesting, because given with IV treatments, we could show that in the animals with CNS affection that we did with the cytostatic, the cells homed to the brain by themselves. And we are exploring the key mechanisms of this behavior at present, but looking at the neurodegenerative states. Also, we are a part of a consortium, one of 4 parties, where we are the project coordinator in an allogeneic NK cell project. It's a long-term project. It's difficult technologies, but we are very proud of being involved in this. We see many possibilities for our core technologies, and we also have a patent portfolio within hematological development and specifically NK cell differentiation in these type of novel systems. And just to show you that this is the development plan for the IPS, and it's a 3-year plan just to bring us to a preclinical candidate, and we are at year 1 now. So these novel systems have many challenges, but we want to be here to be able to contribute with Amniotics technology in building these new products. This is our leadership team, our management group, myself, Gerton Jönsson, Jan Taltz, our Chief Operations Officer, and Andreas Herbst, our Chief Medical Officer. And here is our board of directors with Peter Bühl at the helm and Fredrik Tiberg, Ingrid Atteryd Heyman, and Christopher Bravery, our stem cell expert. Thank you very much for your attention.
Okay, thanks for that. I think the most obvious questions to start with is this about the potency of your neonatal cell. How important is that, since most of the companies in these sectors, they use adult cells, or for example from liposuction?
Yeah, we believe this to be very important. Of course, you can extract adult cells, and you can even have, I guess companies have ways to improve their efficacy, but it is a general fact that the younger the cell is, the less deteriorated it is by external factors, viruses, I mean all sorts of aging. And if you look at the healing properties of a newborn, or as when you were a child, how a wound healed, or when you're an adult, that's basically the answer of the dynamics in tissue. So the younger the source material is, the better. But then there is, of course, the cutoff where you can't be unethical when you source this material either. So if you can have a neonatal material, but without having any ethical problems, then I think it's the optimal.
And as far as I understand, you're the only one using amniotic fluid.
Yes, from the term from a normal pregnancy and using this material during a cesarean section. Yes, we are the only ones that we know of.
I'd also like to focus a bit on this this animal experiment with pigs, because I think the data you've shown are quite exceptional. You're using human cells in animals. Is that correct?
Yes, that is correct. And that is quite surprising, really. When you go into this business, you think that, what? Shouldn't we have pig cells and treat the pigs? But these cells are interesting because they are immune evasive. So I can give my mesenchymal stem cells to you, and you can give yours to mine. And they will do their effect, and then they will disappear, but they will not be confronted by our immune system. And surprisingly enough, this works cross-species barriers. So we can try these cells in rats, in mice, and in pigs, and they still have the biological results. And so we find that it's amazing that it works, and increase. I mean, this gives us confidence that it will work intra-species too.
You might argue if it works that good in pigs, it should work just as well in humans.
That's, yeah.
So and that brings me to the next question. How similar is the clinical study you're planning? Could you talk a bit more about that in lung transplantation?
Yes, the study we are planning is a phase 1 study in 6 patients at the Swedish University Hospital. And it is planned to be a two dosing of the recipient, first in the early phase and then within 24 hours. The animal experiments had an extra layer because we used an equipment similar to what ex vivo has, when you circulate the organ outside of the body. So we actually gave stem cells to the lung too. In the phase 1 study, it's a safety study at first to establish us in the area. We do this because the patients are so multi-sick and have multi-pharmaceutical treatment. So we have to, even though we have safety in another cohort, this is such a particular group. So there is a difference, and I would say that the goal would actually be to pre-treat the organs too. The problem is that the penetration of these ex vivo perfusion equipments is about, at best, 15 to 20%, as I understand it today, which means that with it at the university hospital, 20 to 25 transplantations per year, it would take a very long time for us to do the study. And so from that respect, time being money, we would rather do a safety study first. And that is also what the key opinion leaders say. Show safety, find signals in this cohort, and then develop it finally in the phase 2.
As I understood, it would be a quite cheap study for you.
Yes, investigator-initiated trial. That means that a doctor comes to us and says, I have tried your cells. It's so good in my experiments. I want to try it on my patients. And that's the best way as a stem cell company to operate, because it lowers costs dramatically. I mean, there are costs we have to support, of course, but it's dramatically lower than going to the so-called CROs, the clinical research organizations that is supposed to find the doctor, find the clinic, build the organization around this. So this is the way we want to go with all our product candidates initially.
Absolutely makes sense for smaller companies in your position, definitely. But could you say something about potential partners? You mentioned ex vivo. To me, that sounds like the perfect kind of partner to work with.
I would love that. So I think that the companies that are in this, in Europe we have ex vivo perfusion, their American competitor Transmedics that have a similar system and a good US market penetrance. I think that layering their sort of flushing and maintenance of the organs with the cell therapeutic is the future. So we of course look that and make sure our systems are compatible with the filters, the circulation devices, etc. That is important for us. And I think that this is the way forward, to have good mechanical solutions, chemical solutions, but also cell therapeutic solutions to improve outcome in transplantation.
Yeah, and you will have more after this phase 1 studies conclude, you will perhaps have more leverage in conducting discussions. But anyway, the time is up. So thanks for coming here and presenting your company.
Thank you very much.